So students, this is our experimental setup that we were talking about. As we can see, the experiment is very well written there. It is to demonstrate germination of seeds and the factors involved in it. This particular topic has been taken from class 6th, subject science, topic is food, and the activity that is being performed here is germination of seeds. Now the point here is how to do an experiment or how to proceed on with an activity. We have to follow a scientific methodology. We have to first make our hypothesis, make our experimental statement. Then we have to mention the materials required to do an experiment. Then we have to proceed on with the experiment, that is setup. And finally we have to make down the observations. And in the observations, we have to make down the factors first. How many factors are involved here? Factors involved. There may be physical factors, like here it is mentioned water and air. Then there may be some biological factors involved, like the size of the seed, hardness of the seed coat, and anything that we observe extra, that is structures coming out of the seed. It would be whatever structure is coming out, then we have to check the appearance of that structure. First of all, when does it appear? Then what is the color? What is the size? Whether we can find out any change in the size over the time, and any hair-like growth. Similarly, we will be using the samples, and we'll be arranging those samples as it has been shown here. This table corresponds to 4 columns: column 1, column 2, column 3, and then the column 4. The 4th column we'll be not using at present. Actually, the 2nd and the 3rd column have our experimental setup that involves sample A and sample B. We have kept sample C also ready under the same settings, but the seeds are different. So let's start the experiment, and first we go to the experimental setup and the materials that are required here, so that we can understand how an experiment is done, how an activity is done. Here we can see that to find out the germination, we require materials. The first material, of course, is the seeds of any type. We can take 10 seeds. Then some glassware is required, some petri dishes, watch glass, dropping bottle, and the cotton is required. Now, how to proceed on with the experiment is very important. It's a procedure. We'll first take 10 seeds. Here we have taken kidney beans. Okay, and it could be anything, even gram also. We divide them into 2 groups of 5 seeds each. We have divided there into 5. 1, 2, 3, 4, and 5. Similarly, it is 1, 2, 3, 4, and 5. So after dividing them into the 2 groups, we will take the watch glass. We will place the cotton on the watch glass. Then we will put 5 seeds each on the 2 watch glasses and label them as A and B. We'll add water, little bit of water, in the sample A only, so not in sample B. Now we cover the watch glass. This is the start of our experiment, and we label it as day 1. Now we have to observe the experimental setup for 5 days, and we have to record this, whatever we have observed. Our recordings will be as follows. Now this is our recording. We'll just focus on to the 2 experiments. setup A and setup B. So this was our sample. We already know about it. Sample A and sample B. We'll be focusing on sample A and sample B, which is kidney beans, 5 seeds on each side. We have set up the experiment. This table for sample A has been divided into 6 days. Similarly, sample B has also been divided into days. Factors are written on this side. Now let us go to check the factors on daily basis. So we come on to the observation criteria number 1, that is physical factors. The first factor that we have taken is water. Water has been added or not. On the day 1, sample A, day 1, water has been added. Similarly, in sample B, day 1, water has not been added. So here the variable factor is water present and water absent in sample B. Air, of course, air is a common factor. It is present on first day here and in first day also there. Since the seeds were dry, so the size of the seeds, whatever was the size of the seeds, we have to take it on. Day 1, there is no change, that is same size. For sample B also same size. Hardness, since the seeds are dry, so on day 1 the seed is very hard. Similarly for sample B also, it's very hard. Seed coat, since the seed was dry, so the seed coat was complete. Similarly for sample B also, complete. Structures coming out of the seed. Since the experiment was started on day 1, so naturally there was no structure visible on day 1. Same is the case with sample B. Now since no structure is visible, so the criteria for appearance, color, size or hair growth, there does not come into play. And same is the case with the second. Appearance, color, size, any hairy growth or any leafy growth. That doesn't come into play. So the day 1 data is complete for both the settings. Day 2, what we observed was, again, there was little dryness in the samples A and B because it is hot climatic conditions. So little water was added. Water was added in sample A, but it was not added in sample B. So sample B still remains dry. Air is a common factor. Size, as we check the seeds were swollen in sample A, whereas they were same as before in sample B, no change. Hardness, on day 1 it was hard, whereas on day 2 it's soft. We can feel the softness. Whereas in case of sample B, day 1 it was hard and day 2 also hard. Coming on to the seed coat. The coat was complete on the first day in both the cases, but on the second day, sample A, the seed coat was softer and it cracked. Whereas in sample B, it was still complete. Still there is no structure observed, which is coming out of the seed. So the next criteria of appearance, color, and they all go blank here. So this is the second day, which is over. Today being the third day, we have again watered the sample A. We have watered sample A, but again we have not watered sample B. It is still dry. Okay. So we have to, so our focus is water, availability of water. Does it have any effect on the germination? Okay. We continue with the size. The size is still swollen, whereas in sample B it is same. Hardness is reduced, that is more softer. This sample B is still harder. Seed coat has broken now, and we'll be now checking out the various parameters which are required here.